So our next speaker today is Pat Byrne, who is a professor emeritus at the Colorado State University in the Department of Soil and Crop Sciences. So, all right, thank you, Peter.
Okay, good morning, everyone. Thank you, Vivian, for the introduction, and thanks to the organizers for the chance to talk to you today. It's so great to be here, to see so many old friends and meet new ones, and to do it in person. It's a novel experience. So yesterday we heard from Don Blackburn saying how much he's enjoying retirement. Well, my variation on that is how much I'm enjoying semi-retirement, because I get to stay involved with satisfying projects like this one, but I also have a lot of free time to do those hiking and other things that Don mentioned. So let me start out by saying I'm just a spokesman for this activity this morning. There are many people involved in developing these GRIN resources, especially at these institutions here, Colorado State, Iowa State, and the Ag Research Service. So this is the homepage of grin-u.org. It's a repository for learning materials about plant genetic resources. So many of you are familiar with GRIN. You heard from Peter, and I'm sure you use GRIN services. This is a new component that you can reach through the GRIN website. -U is meant to represent university, so indicating that it's an educational site. So I'm going to come back to this homepage, but first I want to give some background, because this initiative is very much related to this organization and this meeting. So the meeting we had in Pullman in 2015, it was recommended that the PBCC do an analysis of the germplasm system and especially its utility to plant breeders. So a number of us worked on that, resulted in a review paper in Crop Science. And some of the recommendations in that paper were that there was a need to close the gap in gene bank collections, especially crop wild relatives, increase the phenotyping and genotyping activities, pay increasing attention to pre-breeding, improve training opportunities, and expand outreach efforts to the public. So a lot of these are very tall orders that aren't easily accomplished, but my colleagues and I thought that these last two were an area that we could make some headway on. So we developed a strategy in 2018 and 19. We had a workshop in Fort Collins that some of you attended. Thank you for that. And we discussed things like what types of materials would be useful, what format, what topics, how should something like this be funded. And then we followed that up with an online survey, and we got over 400 usable responses, which we felt was pretty good. And we asked that same type of question to try to get a sense from the community of what materials were people looking for. So as a result of that, we achieved a consensus that what would be useful is a versatile set of interconnected learning resources, things like videos, infographics, ebooks, as well as university-hosted courses. The target audiences we had in mind were gene bank employees, and Peter referred to this, the need to train the new generation of people that work and manage gene banks. Undergrad and graduate students, so materials that could be incorporated in courses, and then as well as the general public interested in food and agriculture. So it really took off when we established a collaboration among these three entities and especially when we got a Higher Education Challenge Grant from NIFA. So it started in 2020. We're now in the third year of a 3-year project and hope to keep it going. So back to this homepage, there are a number of ways to search for materials. I find a useful one to be under training formats, and you can see the types of materials. There are ebooks, courses, webinars, videos, etc. So if we start first with ebooks, this is one that we did on crop wild relatives. We like the ebook format because it's very flexible for incorporating text, video, images, and the platform we use is called Pressbooks. Makes it very easy to design an attractive ebook. So one of the chapters is a case study on sunflower, very relevant to our location here in Ames. And some of the things in that ebook, there's a map showing distribution of wild sunflowers. There are images on some of the species. And I would like you to listen to what Laura Merrick had to say. We interviewed her. She's the curator for sunflowers here at the Ames station. And I think you'll see her later today. So.
So sunflower is an unusual crop because sunflower breeders have been using the crop wild relatives to source useful traits since formalized breeding began in sunflowers in the early 1900s. Sunflower as a crop was developed in Russia and Ukraine, so it was domesticated in North America, but crop development took place in Europe. Other disease resistances and tolerances have been provided by wild species for rust, for downy mildew, and Phytophthora, other diseases. In addition to Helianthus annuus has provided some drought tolerance for the crop.
Okay, and then doubled haploids are an important part of plant breeding. I'm sure some of you, at least, are working with doubled haploids. I found in my teaching that it was something that was hard for students to understand and really appreciate. So we did a video on doubled haploids.
In the 1990s, plant breeders began working with a new technology to develop breeding lines in a shorter time frame. This was known as doubled haploid plant production. Since then, this has become a valuable tool to plant breeders across the world. But what exactly is a doubled haploid? The term haploid refers to a single set of chromosomes. A plant's gametes are haploid cells, and when they unite in fertilization, a diploid embryo is formed. If instead the chromosomes in one of those haploid cells can be doubled, a doubled haploid organism is produced, where both chromosomes of a pair carry identical alleles at all the genes.
After we produce the doubled haploids, we bring them out to the field. So generally we get about 10 to 20 seed for each doubled haploid. Take that out to the field and we plant it as a small 2-row plot. And this 2-row plot allows us to produce enough seed to go into replicated yield testing the next year. And so in this 2-row plot, we're looking for a number of different things. So just general agronomic traits. So how tall is it? When does it flower? When does it mature? Is it producing, good spikes? Is it producing a lot of tillers?
Okay, another thing we wanted to do was to provide a virtual tour of the gene bank in Fort Collins, the National Laboratory for Genetic Resources Preservation. They get a lot of requests for tours, but they just can't handle all of them. So we did a series of videos that comprise a tour, and I'm going to show you segments of the first two of these.
More than a century ago, the United States Department of Agriculture established a system for collecting and preserving crop varieties from around the world to help support U.S. agriculture. Here in Fort Collins, Colorado, we maintain this collection to provide long-term storage for all the 20 locations that make up the United States National Plant Germplasm System. Approximately 7.4 million samples, representing more than 16,500 plant species, are currently conserved in 1,750 gene banks worldwide. About 2 million samples are estimated to be unique. They're part of a worldwide effort to conserve, characterize, and use biological diversity to improve the human condition.
Under the red roof of NLGRP, one of the world's largest collections of agricultural plant biodiversity is securely stored. We have almost 1 million accessions representing 10,000 taxa, and most of this material is stored as seed. Before seed samples are placed in cold storage, they need to be carefully processed to ensure they remain viable for as long as possible. So why do we store seed at such low temperatures? 2 factors influence seed longevity and storage: moisture and temperature. Generally, seeds last longer in cooler and drier conditions than wet, warm conditions. We dry seeds to an optimal level, and then by storing them in cold conditions, we can slow down the metabolic activities that lead to chemical reactions that cause seeds to age.
Okay, I'm going through these pretty quick, but I want to give you a flavor for what we have. So we did a series of crop-specific videos. Potato, sorghum, and cowpea are some of those, and I'll show you just a bit of those.
Plant geneticists are also working to create varieties that can better adapt to new diseases.
We are transferring plant material all over the world, and so we are also transferring diseases all over. And we see new things popping up all the time. There's at least 70 major diseases we know of potato. In my research lab, we work on bacterial and viral pathogens of potato, and we're primarily interested in how plants resist these pathogens and in finding ways to help farmers manage the diseases. And so if you can breed a resistant plant, then the farmer doesn't have to spray pesticide, and that saves for everybody. It saves the farmer money and it saves the environment.
Cowpeas are renowned for their ability to thrive under stress, in hot weather, poor soil, and with very little water. These are conditions the world is going to continue to see more of as the impacts of climate change continue to grow.
The global diversity is a big strength for this plant because of its adaptability. With that much genetic diversity and that many scenarios in which it can be grown as a primary food source for protein, the potential for cowpea is always going to be there.
Sorghum is one of the most important cereal crops for global food security. It was first domesticated around 6,000 years ago in Ethiopia and Sudan. It then spread across Africa, into India and the Middle East, and eventually made its way into Eastern Asia. It arrived to the US in the 19th century.
My colleagues in West Africa, one of the things they're working on is trying to take the best qualities of some of the modern varieties that they have been working on and combining them with the best qualities of the traditional varieties that have been grown in that region for thousands of years.
Okay, and then I wanted to show you our most viewed video. We did one on the Three Sisters crops, corn, beans, and squash. The artwork I really love. This was done by our illustrator, Catherine Chen, and we liked it so much that we used it on our bookmark. And there are these around the meeting today. I hope you can find them on one of the tables and take a bookmark home with you.
Have you ever heard of Three Sisters gardening? This practice is a historic gardening or agricultural planting scheme in which certain crops are planted in close proximity to one another. By planting in this way, the different crops not only improve their own health and yield, but also that of their companions or sisters. These benefits are not only expressed in the current growing season, but in the next season as well. Who are the three sisters? The short answer is corn, beans, and winter squash or pumpkin.
Okay, another type of format we have are infographics. This one generally about what gene banks do and how they're used by plant breeders, been translated into a number of languages. Success stories are part of the story we're trying to tell. If you have a success story of ways in which gene bank accession was used in a plant breeding program, we would love to talk to you and try to develop something from that. Really want to point out the courses. These are going to start very soon, August 22nd. There's still time to register. 3 courses, each 1 credit, graduate level courses being taught by Jeff Morris, a sorghum geneticist, and Gail Volk. It works for NLGRP, a specialist in cryopreservation and conservation in general. So if you're interested in this area and you would like to sign up for the course, please check that website. So current and future activities, we intend to publish ebooks on orphan crops and on the impacts of climate change on genetic resources, compile more success stories, complete additional videos to support gene bank operations for new employees, and evaluate the impact of these materials. A new initiative in Colorado, CSU has developed a campus in Denver called SPUR, and it's specifically for food, health, and agriculture, meant to be an outreach site for an urban population. So we received some funding on our project to develop activities for middle and high school students related to different aspects of plant breeding and genetic diversity. So this is kind of a new adventure for us. We're looking forward to see where it goes. I wanted to highlight the 2 grad students working on this project, and they were both here at this meeting. I hope you have a chance to interact with Gina Caramelli and Stephen Gray. And then I want to acknowledge all the people that have worked on this project. Especially want to point out Gail Volk, who has been co-PI and involved in all aspects of the project, and Catherine Chen, who has been our video editor and illustrator. And funding from many sources. NIFA was very instrumental. And we've also received support from the 2 organizations here, from NAPB and PBCC. We have an advisory council of 10 members. Some of you are here at this meeting, so thank you for your service. And with that, I will thank you. I hope you will take the time to visit some of these resources. And
Thank you, Pat. I think we have time for one question. Do you see any hands?
there, right there.
Okay, great.
Hi, Chase Krug. I'm wondering if you are going to collaborate with other institutions on your videos that have larger subscriber counts. So on your YouTube channel, you only have about 1,000 subscribers. Are you going to look toward partnerships with, the public broadcasting service or TED, any of those type of collaborators that could give you video views on higher kind of platforms?
Yeah, thank you. We do have a place on that GRIN website where we list other resources. So there's links to many other videos. But as far as being part of TED, well, we're not there yet. Maybe that would be the next frontier for plant genetic resources at the public.